now we should have a panel discussion, and Dr. Laura Prasad has joined us. And so, are there any questions and discuss points that you want to raise with the speakers in general topics? Yes, I can see a hand going up.
François from Rodis, Rare Diseases. I find it very interesting to see post-authorization safety and efficacy studies for off-label use. In the case that Isabelle Moulon showed, it's from within the same disease, but in other age groups or other genes. But does it mean that off-label use post-authorization safety and efficacy studies could also be mandated for other diseases than the disease for which the drug is authorized?
Good question. Who would you like it to who would you like it to answer it?
Peter Arlett, maybe.
It's a very good question. So there's not an easy answer, is the first point. First, couple of challenges. Firstly, indication, which is, in regulatory terms, the population and the disease where there's an active approval for the product, is not actually clearly defined in European legislation. And that causes all sorts of regulatory problems. So that's problem number one. Problem number two is in the area of post-authorization efficacy studies. We're waiting for word from the European Commission, which will probably be a public consultation on the situations where post-authorization efficacy studies can be places obligations on the industry. So that's another challenge because we haven't yet got clarity on those situations. It's a bit easier for post-authorization safety studies, and here I'll be brave enough to actually try and answer your question. So if you're talking about the same disease, and of course we can have a big debate about how you define diseases, but if you're talking about the same disease, but a different subpopulation or a different severity, and it has been identified in the marketing authorization dossier that there are concerns about safety or even so-called missing information. In other words, that the benefit risk has not been demonstrated in a subpopulation, which might be children or it might be a different severity, just to give two examples, where you will reasonably expect that drug to be used, then it's very clear to me that you can place the obligation on the company to do that study. Now, I know I gave a presentation a couple of days ago on the new legislation to the Pediatric Committee, and you'll know that that committee has had a long wrestle with the new pediatric legislation as to the scope of where pediatric investigation plans can put obligations on industry. And again, these same issues as to what is the same disease, what's the different disease, what is the within the indication or outside the indication come up. But for me, the key point is if it's reasonable that this drug is going to be used in everyday medical practice in a particular severity or subpopulation where you haven't got safety demonstrated pre-authorization, or the benefit risk can't be evaluated, but yet you can expect that use to occur, then yes, you can place that obligation. So that was quite a complex answer. Let me just summarize then. So no legal definition of indication, so we can always debate what is the same disease. For efficacy studies, before we can really start using this new tool in the new legislation, we need legal clarity from the Commission. But for safety studies, we can start right now. Yes, it can go into the margins of off-label use. And the other thing maybe on the off-label use to mention, the last thing, is if we have evidence of safety problems with off-label use, and those problems might even come from a clinical trial in a completely different indication, the new legislation is crystal clear that we can take action even in the on-label use to protect public health. And that's an important change in the legislation, which I didn't highlight in my presentation.
Stuart Hogarth from King's College London. This is a question directed towards Peter, and it's really about the implications for pharmacogenomic relabeling of the new powers that EMA now has. Specifically, you talked about the centralization of data collection for nationally approved products. So I just wonder what the downstream consequences of that are in terms of, for instance, a drug like warfarin, which was approved nationally, which of course the FDA has relabeled twice, but as far as I'm aware, no pharmacogenomic relabeling has been done within Europe. Is EMA now empowered, should the data so support such a move, to relabel a drug like warfarin, which was approved nationally?
The simple answer is the agency, at the request of a member state or the European Commission, is empowered to review the safety of any substance or product on the European market, and the outputs of that review are legally binding. The reason I emphasize is that this I can't wake up one morning and decide that today it's warfarin, and that's quite important in terms of the legal right to initiate an action. So the right comes from the member states or the European Commission. That said, based on the signal detection activities and the routine surveillance that's in place, this agency, and particularly its scientific committees, can make recommendations to the member states or to the Commission to initiate the formal action. And I'm being a bit pedantic in regulatory terms, but if I claim that we have a power which we don't, I get quite forthright feedback from the lawyers after the meeting. So the answer is, if asked to, yes, we can take that action. It will be legally binding. It then means that all product information for warfarin must be updated in line with the new warnings, whatever it be. Now, there's a couple of interesting parts of that, which those of you with a more regulatory bent, I think, will find interesting. Firstly, the classical model is that you have a referral with a legally binding output, and the output addresses the different companies that have got that product on the market, and it says you shall, by such and such a date, update your product information. That either comes from the European Commission or the member states do it directly as a consequence of the review that goes on at European level. One of the things that's really interesting in the new legislation, and it'll be very interesting to see how it pans out over the next couple of years, is that the legislation has a new transparency tool where all of the outputs from this committee are published. And the legislation says that the pharmaceutical companies shall keep their product information, so their labeling, up to date with the recommendations and opinions and decisions coming out of this agency. So all of a sudden, we have changed the legal responsibility on the company, whereas in the past, the company could legitimately say, I haven't detected this safety issue, I'm waiting to be told to update my product information, and I'm waiting for that letter to arrive through the post, which was a very slow and sometimes bureaucratic process. Now there's an obligation on the companies. If this agency, as a result of a recommendation from the new committee, places standard warning for warfarin on the website, there's an obligation on the companies for them to keep their product information up to date. And that should, if the implementation phase goes well, if this really kicks in, and I see no reason why it shouldn't cut out months and months and months of the implementation phase of new warnings reaching patients. Just because you open a very interesting issue. The issue for me, and I think the concept paper on pharmacogenomics is fascinating, but horribly challenging, because the issue for me, we often receive feedback from the Pharmacogenomics Working Party, and it's about what you would do as a clinician with that advice. And I'm sure you struggle with this in your daily practice, and perhaps many of you that are clinical pharmacologists in clinical practice have the benefits of easy access to some of this pharmacogenomic testing. But often, you saw some of the wording on Ying's slides, if you can have access to the testing, or if that you're known to be to have a certain allele, then consider carefully benefit risk. You know, these are really difficult for anybody other than the most specialist pharmacist or doctor to deal with. And I think, the direction of travel is in the right direction, but we have a huge amount to do before this translates easily into changes in clinical practice that will reduce risk and potentially potentiate benefit.
Thank you, Peter. I think in that one sentence, you summarized our 2 years of our work as to how we discussed as to what wording should be there in any of those labels. Magnus, please. Thank you.
In 1998, a very interesting paper came out in the Journal of American Medical Association, which actually define more or less adverse drug reactions as a disease. And if you look at the registered adverse drug reactions, serious adverse drug reactions, coming to FDA, they have increased by a number of about 2.6-fold until 2006 from 2000, whereas the prescription of drugs has only increased 30 to 40%. And to my opinion, there might be two reasons for this drastic increase in registered adverse drug reactions. One is that actually the reporting from the physicians have been improved and better, and the other is that we have introduced biologics into the market, which have unforeseen adverse drug reactions. And I would like to listen to Peter's opinion, which is the explanation for this scenario.
Maybe if I start, I might then bat it to Ying, because I'd be very interested to hear Ying's view. It's very interesting what you're saying. We have a discussion tomorrow morning internally, and then with the member states later in the week, on a strategy to measure the impact of the new legislation. And I've had the unenviable task of drafting this strategy with one of my colleagues, Ulva Spooner, who's the vice chair of the Pharmacovigilance Risk Assessment Committee. Because if you really look at health outcomes, which is the ultimate objective of what we're doing, then that's really challenging to measure, because there are so many variables that will affect actually the level of adverse drug reactions. If you do what most people love to do, and you've just made reference to it, because it's relatively straightforward to do, and just to count adverse reaction reports that come in, it's impossible to analyse what's going on, because, as you've outlined, it could be that there's real change in the safety of medicines on the market. It could be that doctors and pharmacists and patients are using medicines more or less safely. Or it could just be a change over time in the uptake of reporting schemes. And you asked me my view. My view is that by far the most likely is the last. It's just a change in approach to reporting. And if you look, the other thing that I think is very interesting, and the warfarin was a good example, codeine in Isabel's presentation is a really interesting example. If you look at those surveys that have been published many times in peer-reviewed journals of the burden of adverse drug reactions, it's drugs we know about. It is warfarin and non-steroidals and aspirin. It's not the biologicals actually that are doing the really high numbers of everyday adverse reactions that are seen bringing people into hospital. I'm not saying it's not the biologicals, but I'm saying in absolute percentage terms, it's all of the old, well-established drugs with well-established safety profiles.
Yes, I agree with what Peter had said. Spontaneous reporting is very useful for signal generation and to understand the pattern, but it's not a good tool to look at the quantity, the magnitude of the problem. And if we really want to know the incidence or frequency of the adverse reactions, I think we have to do studies or use other tools.
Just a thought which occurred to me, that apart from the 2 points that you raised, Peter, in terms of reporting in frequency and also the number of drugs which is being used, do you think there is a role for or an impact for the polypharmacy that happens, which seems to be happening much more now because of approved labelings and approved indications for the number of drugs used in combinations, for the multitude of comorbidities that patients have? Do you think they have an impact on the adverse reactions?
I think a very quick answer is absolutely. There was a very interesting session at the International Society of Pharmacoepidemiology in Barcelona at the end of August. It was particularly focused on medicines in the elderly and the whole area of polypharmacy, and the fact there were some really good presentations from people that had studied expert guidelines. And basically the conclusion was that expert guidelines are great if you have one disease, but by the time you've got 3 or 4 or 5, you're just lost, and it then comes down to finger in the air clinical judgments. So I think absolutely the more drugs you're on. And there was one other very interesting thing I thought at that session. Again, it was focused on the elderly, but was if you see a new symptom in an elderly patient, it's probably an adverse drug reaction, as you should be your first thought. And I think there's a lot of truth in that. You should rule it out before you start treating the symptom.
Excellent. Thank you. Yes, please, Isabel.
I have a question, but focused on pediatric medicines. How we can address the question of safety biomarkers on metabolism if you know that, for example, CYPs have a different profile of expression during liver development. In my opinion, this question is slightly addressed in the guideline of pharmacokinetics. And I would like to have the opinion of Q, if possible.
Yes, indeed, there are vulnerable populations of the subpopulations. We have poor metabolizers, extensive metabolizers in general population that are at risk for certain toxicities. But within these groups, you can still have renal disease, liver disease, or use other drugs that inhibit the alternative metabolic pathways that make these patients are more vulnerable. In addition, if you have elderly or pediatric patients that have immature organ development, that makes these subpopulation even more vulnerable. So I think these will all be addressed in different guidelines and should be taken into consideration.
Dr. Spielberg?
As a pediatrician, I have to weigh in. So, but there are some very well established examples where there are polymorphisms in drug metabolism in the adult population. But if you look at the ontogeny of those pathways, they disappear. Recent studies on CYP2C19, where clearly there are people who are rapid and slow metabolizers in the established population, you move into the less than 1 year olds set, and basically they're all slow metabolizers. And it's difficult to, you have to really map out the ontogeny of each individual pathway to know if those genetic polymorphisms in fact are represented in an ontogenetic sense. And often they're not. Sometimes they are, because in fact the enzymes are fully expressed at birth, and you do see the polymorphisms. And it's very much pathway dependent, and individual CYP, individual phase 2 enzymes, etc.
Excellent. I don't see any other hands going up, so it leaves me to say thank you to all the speakers and to the chairs for the day, and specifically to thank Dr. Moulin for stepping in as the third speaker for this session. Thank you, Peter and Chunying, for an excellent discussion on the regulatory aspects and what the new regulation brings about. I will try and summarize the day in the next 5 minutes. If I fail, I was the one who overstepped the mark at the end of the day. To begin with, I want to say thank you to all the people who made themselves available to attend and speak at the conference. And this workshop is only as good as the people who attend it, and you all made it, I think, a very good first day for me. I have to thank specifically the number of colleagues and friends who have had the pleasure to work and collaborate with, who have traveled far to be part of this effort, and their presence is very much appreciated. In the first session, I think we began with an excellent talk about genomics and clinical care. Should I say the French way, which is a very good model, I might add, for oncology. Dr. Nowak showed us how to get multiple centers to work together and deliver genomic-based treatment successfully in a large group of patients. I think she pointed out the organizational excellence in a combination of scientists, clinicians, and the local government organizations, and I think it's a lesson for all of us to learn. If I were to judge the session by the number of questions raised, it's evident that this was very popular and actually very much appreciated. And I take this opportunity to thank her for her efforts in being here today. Next, we heard from Federica Goodsed about cystic fibrosis and molecular phenotype and a novel concept of N=1 trial. And I must say that this was before I had heard him before, and I'm always impressed by him as a speaker. And he brought out the crucial aspects of rare diseases and how do we tackle them. And from a regulatory viewpoint, this is a point of discussion. I think, in my mind, we haven't handled it very well so far, and I think we still need to get some way forward to address this. In terms of making this N=1 number trials as a reality, or even getting the rare disease treatments to patient populations, I think we have a fairly hard task ahead, not just for regulators, and I think it's for everyone, the industry, the clinicians, the government organizations, and HTAs. I think we ended up with a discussion on criteria for rare diseases and orphan diseases, and I'm glad to say that this is an open and debatable point. And in terms of what we consider as rare, if it doesn't meet the numerical values that we set ourselves, does it imply that it is not rare? Or how do we tackle it? And I think it's a question for us to address in the future. We then heard from two excellent speakers, Mark and Michael, who talked about pharmacogenomics and PK studies and early phase studies. And as you will have noticed, that there are some interesting differences between the way those two speakers approach the issue. And it is for us to see if there are common grounds that we can take forward, appreciating that they were trying to address two different aspects or two different points. One issue which I would not want to really discuss much about is the aspect of companion diagnostics and how we're going to tackle it. And that is a difficult one at this stage, and I think it's outside the scope of this meeting simply because of the implications that we have, or it will have, in a number of areas. I can only apologize for cutting short the question. It wasn't intended to be to stop the discussion, but it'll come up tomorrow, and we will try and discuss it then. The second session was chaired by Rob Hemmings, opened up the methodological aspects relating to confirmatory trials, and there were two excellent speakers who brought out all the answers that you wanted to know and not agree with. I say that as a clinician. I think that these points have been discussed a number of times. Whether we agree or not agree, there are issues that we need to tackle in terms of the statistical and methodological issues. But I think Martin Post did a good job of giving us some hope in how to adapt clinical trials and take us forward. And Dr. Devanand warned about how assay sensitivity and differences in assay capabilities will hinder some of those approaches. This brought up to the discussion about need for smoothing out the differences in different regions and the harmonization of viewpoints in how you interpret and the need for enriched trials and when to do enriched trials and how to tackle them and how adaptive can you be in designing your clinical trials. Maybe this is an opportunity for all the three regions and regulators in the three regions to sit down and discuss, is there a probability that we would find some common ground and find something which we can all agree to in terms of facilitating the clinical trials, especially for rare diseases, and using the adaptive designs. It's also fair to say that the panelists in the audience, including, and first of all me, that we were stumped by the need to consider pharmacogenomics of placebo response, which Dr. Rina raised. I have absolutely no idea how to deal with it. All I can say is that let me have that placebo. Coming to the last session, I think Peter very eloquently pointed out the update to the regulation and opportunities and improved safety evaluation. He highlighted why we needed the change and how this opens up research opportunities for all of us, industry, academia, and regulators, and how gives us an option for collaboration. And I hope you're all taking notes and will make use of this by accessing the vigilance database in about 18 months' time. Is that right? In about 18 months. Xuning did a wonderful job of trying to tell you how, and she's going to formulate the concept paper on the paper on guidance for pharmacogenomic studies and during pharmacovigilance activities. And she was also kind enough to discuss the drawbacks of the study so far with markers that we have evaluated. And of course, she repeated the oft said caveat that we need to have comprehensive sample collection. Last but not the least, Dr. Mulan did a phenomenal job of stepping in at the last minute, as I said earlier, first as a speaker, and secondly as managing the whole session with giving you the idea of what we are looking forward to in terms of what we can look forward to in terms of the SPC and how to update that. Summarizing, I think there are several recurring themes overall of the day. I believe that they are going to continue recurring unless we all put our heads together to resolve some of them. And I think this workshop has given us a good opportunity to start doing that. And hopefully some of those will be answered tomorrow. Thank you very much, and thank you all for coming and making it a very good day.